Assalamualaikum and hi, I am Hazirah Binti Anuar and my project is under supervision of Dr. Nur Binti Sharizan. The title for my final year project is The Effect of Pre-Treatment with Melastoma malabatricum Leaf Toward the Bitterness of Carica Papaya Shoots and Their Phytochemical Properties. Background of study. Carica papaya are categorized as an edible plant and usually known as papaya. Regardless of sweetness of papaya fruit, the papaya shoot and leaf are bitter. But for a long time, papaya shoot and leaf have been used traditionally to treat various diseases such as dengue, malaria, and chikungunya. Melastoma malabatricum is commonly known as senduduk in Malaysia. In Malaysia and Thailand, senduduk has been used in traditional medicine to cure a range of diseases. On the other hand, in Indonesia, senduduk leaf are often used traditionally to mask the bitter taste of processed vegetable dishes such as bitter melon and papaya flower. Next, there are two problem statements of this study. First, various measures have been taken to mitigate the bitter taste of papaya shoots, such as by soak in a salt solution before cooking. But these methods still leave a bitter taste in the mouth. Second, there have been only a few published scientific articles on the application of senduduk leaf, specifically on the bitter taste reduction activity. Next, significance of study. The effect of pre-treatment with senduduk leaf toward the bitterness of papaya shoot was studied using the sensory evaluation test. This study can contribute on providing scientific evidence on the application of senduduk leaf to reduce the bitterness in bitter plants. The result from this study can provide information about product differences, consumer preferences, and attribute intensities. There are two objectives of this study. First, to examine the effect of pre-treatment of senduduk leaf towards the bitterness level of papaya shoots via sensory evaluation study. Second, to analyze the phytochemical constituents in papaya shoots with and without the pre-treatment with senduduk leaf. The sensory response to taste have a big impact on a dietary choice and eating habits. Bitterness is 1 out of 5 taste sense and humans are very sensitive to it. Usually, the bitter taste is described as a flavor that is strong, pungent, or harsh. The bitterness can be found in a variety of foods. Amino acids and peptides, ester and lactones, phenol and polyphenols, organic and inorganic salts are all bitter chemicals that can be found in meals. Carica papaya, commonly known as a papaya or papau in Australia, and it is belong to Caricaceae family. Various parts of Carica papaya plant can be eaten such as young leaf, flower, fruits, and papaya peels. All parts of Carica papaya plant have a wide range of therapeutic properties such as antihelminthes, antiprotozoan, antibacterial, antifungal, and anti-inflammatory properties. Melastoma malabatricum is belong to Melastomaceae family. Usually, they are found in the tropics and subtropics, and there are over 4,000 species worldwide. Senduduk has a wide range for medicine applications such as diarrhea, dysentery, cuts and wound, stomach ache, and sore leg. They are also used to distinguish the bitterness of processed vegetable dishes. Phytochemicals are natural chemical substances found in plants. Plants create this compound as a result of their primary or secondary metabolism. Alkaloid, tannin, flavonoids, saponin, and phenolic compounds are examples of phytochemical with bioactive constituents. Some plants have a therapeutic properties due to chemical compounds that have specific physiological effect on the human body. Many of these therapeutic plants are also useful as a spice and foods. Experimental design. Firstly, young papaya leaf or shoots and matched senduduk leaf were collected. All samples were carefully washed with distilled water to remove any unwanted particles. Then, for sample treatment, 100 grams of fresh papaya shoots have been boiled in 1000 milliliter distilled water, while another 100 grams of fresh papaya shoots have been boiled with presence of senduduk leaf with various quantity at temperature 90 to 100 degrees Celsius for 30 minutes. After the sample treatment process, all treated papaya shoots were separated into two parts. One part was used to determine phytochemical constituent, while the another half was used for sensory evaluation study. The phytochemical screening revealed that the fresh papaya shoot sample G contained phenolic, flavonoid, saponin, tannins and alkaloid. Phenols, saponin and tannin were detected in a large quantity, whereas flavonoid and alkaloid present in a small amount. However, sample A showed a moderate amount of phenols and saponin and small amount of flavonoid, tannin and alkaloid. Meanwhile, lower amount of compound were observed in sample B and sample C. In sample D, E and F, phenols and saponin are not present and small amount of flavonoid, tannin and alkaloid are present. This indicates that certain phytochemicals could be removed through the boiling process. Besides that, pre-treatment with senduduk leaf was also suggested that could release some compound which indicate by lower detection through the phytochemical screening. It was found that the treatment of the papaya shoots had an effect on the color intensity of the samples compared to the fresh sample. It is observed that compared to the untreated sample with a darker color, the sample treated with a 25 gram of senduduk leaf in particular had the lightest color intensity. Sample A, B and C show a green color but with a different intensity, contributing to a fresh appearance characteristic of chlorophyll content. On the other hand, the color changes could be influenced by the different amount of senduduk leaf because in this study, the same amount of papaya shoots and volume of water were used and the extraction was monitored at 90 to 100 degrees Celsius for 30 minutes. According to table 4.1, the color changes could be due to the presence of phenolic compound or saponin. It is because the color indicated by papaya shoot extract with senduduk samples D, E and F were yellowish. It may be due to the phenolic compound and saponin are absent. The phenolic compound have important function for the sensory quality of the product and influence the color, taste, smell and aging. In quantitative descriptive analysis result, it was found that among all the 7 samples, the samples treated with a senduduk leaf with an absorbance of 2 grams scored the lowest for bitter taste. As described in table 4.1, it is significant that the presence of phenolic compound and saponin throughout the process was related only to the color changes, while the intensity of bitterness of the samples was not related to the color changes. However, taste panelists were extremely sensitive to the very low levels of bioactive compound in the papaya shoot samples. The threshold for what is or is not acceptable may vary from person to person as individual taste response to the bitterness vary enormously. The finding of this study indicate the presence of various phytochemical in the papaya shoot extract such as phenolic compound, flavonoids, tannin, alkaloid and saponin, which may be responsible for the pharmacological activity. This also influenced the color intensity of the papaya shoot extraction by the presence and absence of phenolic compound or saponins. On the other hand, the bitterness of papaya shoot was successfully reduced by senduduk leaf by using the boiling method. Further extensive research investigation are needed to explore the quantity of papaya shoot bioactive compound after treatment. Determination and quantification of the bioactive compound can be conducted by using high-performance liquid chromatography or HPLC, and color measurement are needed to carry out by using Minolta chromometer.